All right, Dr. Zach Berndsen and the Berndsen lab at Mizzou are focused on biomolecular systems within the context of human disease with a goal of creating bridges between basic and translational science. Zach grew up in St. Louis. This is, by the way, his first time in Kansas City. So, now, while he works at Mizzou, he did not go to Mizzou, but he was very quick to tell me that he's got a lot of people in his family who did, so you're solid with us. Okay. When he's not in the lab, he says he can be found down at the Ozark National Scenic Riverways. He's gone there every year since he was a teen to float, camp, and fish, so it's about that time of year.
It is.
All right, welcome.
All right, thank you so much. Thank you to the organizers, and thank you for everyone showing up. So, I started my lab in the summer of 2022. We're at Mizzou Hall, and we're Mizzou, and we're located at Stevens Hall. So if you're familiar with campus, it's a really beautiful corner of campus, and these are a picture of some of my grad students and undergraduates. We just hired a senior staff scientist, so I hope my grants get funded. A lot of what we do is, and all the data I'll show today was from data collected at our brand new electron microscopy core facility in the NextGen Precision Health Building. And so this is very much a field of dreams situation where if you build it, they will come. So they built this amazing facility, and it's what attracted me to campus, in addition to obviously coming back home. And so just a brief background on what cryo-electron microscopy is. So particularly single particle. We'll start here with purification of your protein or your molecular complex of interest through either cell culture or, as I'll show you today, from human serum. And then we apply a small amount of that sample to a holy carbon grid or gold grid, and that's plunge frozen then into a liquid nitrogen temperature liquid ethane. And the idea here is that you'll have a bunch of individual particles distributed at random orientations within your vitrified ice, which you then image with your electron microscope. And we collect thousands of these images. And then we pick these individual particles, and we use algorithms very similar to how an MRI or a CAT scan or a PET scan might work to reconstruct three-dimensional density maps, which we can then use to interpret and build atomic models. And so we can see here just a zoom in on one of these cryo grids, which is about the size of a pencil eraser. And so this is very much what you'll see in the electron microscope, and you'll have to identify regions that are of interest, and then you keep zooming in, and we'll see these little holes that have been drilled into these carbon films, and it's inside these holes where your protein and your ice will be. So you'll want to image directly through the center of these holes, and as we'll see when we get in, you can see individual proteins or protein complexes. And so what's so great about cryo-EM is that you can, compared to other structural biology techniques, is you have a broad molecular weight range. It requires less sample, and particularly in my cases, you have this ability to deal with sample heterogeneity. And this was resulted in the Nobel Prize in Chemistry in 2017. So my lab focuses on a lot of different projects, from structural-based vaccine design, from viral pathogens and bacterial pathogens, to very basic structural biology. And so what I'll talk about today is our recent published work on lipoproteins. And so lipoproteins are large and heterogeneous macromolecular nanoparticles. They're composed of a phospholipid and free cholesterol monolayer with these embedded apolipoproteins. And there's a core of triglycerides and cholesterol esters. And they can be classified by their density and their size. And so we have these chylomicrons, which are made in the gut after you eat a fatty meal. And then we have VLDL through LDL, which are produced in the liver and are the primary transporters of cholesterol and fats in your blood. And then we have HDL. And they have each these non-exchangeable lipoproteins. So these are the main structural components. And we see here what I'm particularly interested in is apolipoprotein B100, which is on these three different species. And so what happens is this protein is made in the liver, and as it's coming off of the ribosome, it starts sequestering fat molecules by interacting with these chaperones to form this nascent particle, which then traffics through the Golgi, emerges with these lipid droplets to create this very large triglyceride-rich VLDL, which is exported into the bloodstream, and the triglycerides are delivered to the peripheral tissue for energy through the conversion to free fatty acids from lipoprotein lipase. And during this process, the particle is shrinking in size and increasing in the content of cholesterol, which this LDL particle, which a lot of people know about, which is then cleared from circulation by interactions with this lipoprotein lipase, with the LDL receptor on hepatocytes. And so ApoB has these three primary functions where it coordinates the synthesis of this protein, of this particle, It maintains structural integrity over this entire process here, and it also serves as a binding partner for the receptor, allowing it to clear from circulation. And the reason this is important in human disease, of course, is that LDL is the primary driver of atherosclerosis. And so you go to your doctor, you get your blood drawn, they tell you your cholesterol is too high. Really what they mean is there's too many of these particles floating around in your blood. And we see that cardiovascular disease, in particular atherosclerosis, is the leading cause of death worldwide. And in the Midwest and in our region, it's a particularly bad problem. And so revealing the structure of this integral protein is going to kind of create a foundation for the development of next generation therapies for more targeted treatment of cardiovascular disease. And so the reason this was such a challenge is that ApoB is one of the largest proteins in the human genome. People have been trying to understand what this thing looked like for 50 years. And you cannot separate it from its lipoprotein particle. It's extremely heterogeneous, and there was no previous structures of this. So cryo-EM is uniquely suited for tackling these challenges due to the reasons I mentioned earlier. And so this is our general workflow. We can take blood from a patient, and this is really powerful because we can have a patient with any phenotype here. Could be a healthy patient, could be one with diabetic dyslipidemia, atherosclerosis, or known genetic disorders. We can then purify with an ultracentrifuge the lipoproteins from their blood. And then a further step that I do in my lab is we'll take these LDL fractions and then we purify them over gel chromatography to separate them based on size. And then we can image them on our Titan Krios electron microscope at the NextGen building. And so this is just showing an LDL sample that we have here run over a size exclusion column. And it's a nice sharp peak. And so what I decided to do for this initial experiment was to take these smallest possible particles. And there was several reasons for wanting to go after these small particles to begin with. And we collected about 13,000 of these large images on our electron microscope. And we can see here these individual LDL particles. And after a significant amount of processing and GPU compute resource time, I arrived at this high to medium resolution reconstruction of what I believed was ApoB100. And so this is by far the best picture anyone have ever had of this protein. And so we kind of saw that this globular N-terminal domain and then this belt of high density that wrapped around the exterior. And then at these low contour levels, so that the weaker intensity data, we can see that there's this liquid crystalline core actually of cholesterol esters. However, I was able to kind of do additional computer processing to kind of find subsets of my data that don't have this ordered core, and so we can start to begin to look at how the content of the core affects it. Given no previous background data on this, we used AlphaFold, which was another Nobel Prize in chemistry, which is an AI algorithm for predicting protein structures. But the problem is it's really good at globular proteins, but this thing sits on the surface of a big fat ball, and so the AlphaFold didn't really know what to do with it. The local structure was great, but the global structure wasn't. And even the newer iterations of AlphaFold really rolled this thing up like a blanket. And so we used the GPU computing resources available at Mizzou to run large-scale molecular dynamics simulations that take the cryo-EM density as an additional biasing potential in the force field and really pull the protein structure into the cryo-EM map. And this is showing the fitting of that beta belt, and this was done by my colleague in the physics department. And these other domains of the protein required more sophisticated efforts that I won't get into here. The end result is this beautiful structure of full-length ApoB100, and this is about 5,000 residue protein, so really huge. What we see is this globular N-terminal domain with this kind of structure, and anywhere that's inside the center of this cage-like structure is amphipathic, meaning it's bound to fats, so it's bound to the interior triglycerides or cholesterol or surface phospholipids. And what we discovered is it creates this gigantic beta sheet that wraps around the exterior of the particle and holds it together like a belt. Interspersed within this belt are these different 9 inserts. Some of them are quite large, as much as 700 residues, and they reach across the surface of this lipoprotein particle and make really interesting strategic long-range interactions. And we believe that this helps hold the particle together and coordinate these interesting conformational changes that I'll talk about. And so just a few of the things that we can learn from this structure is we can start to map all these interesting genetic variants that people have that result in either very high in the case of FH or very low in case of FHBL levels of these lipoproteins. And the patients with FH mutations will have early onset atherosclerosis. We can also begin to propose a structural model for how this protein might change shape as the size of the lipoprotein particle change. And so we made this hypothesis, and we published this data in Nature magazine in December, and we've been getting a lot of great press on this. So to the students in the audience, it's not about where you publish, but it's still fun to publish in Nature. So the next step, so this is all unpublished now, the next step is we want to start looking at these larger particles just in this LDL fraction. And so we took the other end of our size exclusion peak, collected a bunch of data on our brand new electron microscope after our core flooded. We got a brand new one. We can collect more data even faster, so we made 50,000 micrographs of 2 different fractions. This is the size distribution of the particles, just in blue, showing that the particles we're now collecting are larger than the ones I just showed you. And then we have these new reconstructions of this particle, again, with and without this ordered core, so we can start to make sense of how the changes in the core structure are affecting the protein. And we can also begin to see really interesting ordered lipids. So the lipids are binding the interior of this protein, making all sorts of interesting lipid microdomains. And my colleague in the physics department is running all AtomMD simulations with these bound lipids now. And so we can start to map these conformational changes as the protein grows and shrinks. And this is really a small size range over which this protein can accommodate these particles. And so this is just kind of the beginning of this phase of my research, but a lot of what we predicted is coming true. So the next part of what I'll show you is we're also interested in its interactions with the receptor. So we're done? Okay, so basically ApoB binds the receptor here. It gets endocytosed. The pH change triggers the breakdown of this particle. And so we wanted to look at the structure of this. And so we expressed it in the lab recombinantly, complexed it with LDL, and we solved these two structures at the different sizes of this particle bound, and we found 2 copies of the receptor at different binding sites. And so one really cool thing is we can start to map these disease-causing mutations and how they affect the interactions with the receptor. And another thing we discovered is that the receptor will dimerize this particle, and there's actually this really interesting size-dependent effects where where small LDL particles are cleared from circulation much slower. And so what we discovered is that this dimerization occurs at a very small subset of particle sizes, and this is probably what triggers the endocytotic event. And so with that, I'd like to just really thank the director of the Mizzou EM Core, Dr. Min Soo, and his staff, Reja, and then the members of my lab, Mizzou IT, my collaborator in the physics department, and my startup package. Thank you.
Thank you. All right. We're going to start with you.
Okay. Thank you, Zach. We're really excited to have you in the building. Can you explain some limitations or drawbacks maybe that you face with EM imaging?
So honestly, it went really well with this particle to begin with. I was actually quite surprised. The main limitation is because the sample is so heterogeneous. We collect maybe a couple million particles from all of these images, and the final reconstructions that we get are from maybe like less than 10% of that. And so collecting enough data, storing that data, I mean, I have, 50, 60 terabytes of data, and then processing that on the GPUs becomes really computationally intensive. So that's a major limitation. And then we want to start looking at these larger particles. The limitations of single particle cryo-EM are really, we're hitting the wall on that, and so we might have to start using more advanced technology called tomography, where we collect tilt series of these images and reconstruct these lower resolution tomograms.
No? Okay.
Great work. Congratulations. I was wondering, you mentioned a little bit that you were looking at the lipids inside the particle, but I was wondering, what about other molecules like maybe sugars, glycosylations and maybe cholesterol? Do you see any of this?
Yeah. Well, the cholesterol is part of the lipids, and so these simulations that we're running have the cholesterol in the core. And so we're observing, it's the early days of these simulations, but we're observing some really interesting stuff there. ApoB is glycosylated pretty heavily, and so we can start to look at that, and some of that glycosylation affects its interactions with matrix and these kind of things. And so that's definitely an area where we want to go after kind of doing this initial structural work.
One more question. Yes.
Hi, my name's Adrian. I'm a second year at
Adrian.
Mizzou. First of all, thank you so much for your talk today. This is amazing, of course. Now, from a clinical perspective, current guidelines for controlling things like hypercholesterolemia, dyslipidemia within the context of cardiovascular disease are statins. You know, we memorized this mnemonic in med school called MODA-BASH, where the S in BASH stands for statins. And moving on to the potential like pharmaceutical implications of your research, you've mentioned potential mutations that may cause atherosclerosis, potential endocytotic or like dimerization processes that may contribute to atherosclerosis. What are some of the ideas you have, or perhaps processes that you were thinking about targeting when creating drugs to potentially further control dyslipidemia?
Excellent question. Yijin was one of my medical students in my PPL class, so really good to see you. Yeah, so these statins are obviously very effective, and we have these PCSK9 inhibitors, which are these monoclonal antibodies. And the thing about statins is they kind of shut off cholesterol synthesis all over the body, and it leads to a very indirect feedback network that lowers LDL levels. And so people have been trying to explore the use of maybe monoclonal antibodies, but you're always limited in your ability to design those without structural work. And some of the stuff that we're finding is with these conformational changes. A lot of people in the field think that these really small, dense LDL particles are actually more atherogenic due to their weaker interactions with the receptor, their longer residence time in the blood, and their ability to diffuse to the endothelial wall easier. And so what we're discovering is that as the particle shrinks, the conformation changes. And so now that we understand the very intricate 3D structure of this on these small particles, we could potentially design more advanced targeting monoclonals or bispecifics that could specifically clear these very small atherogenic particles. And then people that might have mutations that inhibit the binding of the receptor, you could think of maybe designing some kind of, maybe using AI tools now that we have to design specific binding proteins to kind of latch the two onto each other. Now that we have a structure, we can use that as a template to design it, which would then help the particle stay bound and be cleared from the circulation better.
Thank you.
All right,
Thank
thank
you.
you, Zach.
All right.